Dr. Devin Gaggar from One Alphabet team, and it's my pleasure to welcome you all at today's session. Welcome to the webinar topic named Innovation, Evidence and Access. Can a clinician have it all for semaglutide? As the burden of diabetes continues to rise with a significant proportion of patients also facing weight-related challenges, diabetes has emerged as a critical clinician concern. In this evolving landscape, innovator semaglutide has transformed the way we approach glycemic control and the weight management. With the current market surge, it becomes increasingly important to understand what truly differentiates therapies, ranging from manufacturing expertise and clinical evidence to real-world experiences. Now, today's discussion aims to bring these perspectives into sharper focus and support more informed, evidence-based clinical decisions. To take this forward, we are joined by an esteemed panel of experts, which will be moderated by none other than Dr. Mala Dharmalingam, Director and Head of Department of Endocrinology, DEDRC, and our esteemed panelist, Dr. Ganesh H.K., Professor and Head of Department of Endocrinology at AJIMS, Dr. Joe George, Senior Consultant Endocrinologist, Director at EndoDiab Group of Institution, Dr. A. Lakshmi Lavanya, Managing Director and Consultant Endocrinologist at American Institute of Diabetes and Endocrinology, Madhapur, Hyderabad. I hand over the session to you, Dr. Mala.
Thank you, Dr. Devan, and a very good evening to everyone. Today, we are in a very interesting crossroad where semaglutide's usage has become quite more compared to what it was. So let's look at some of the emerging data and we will deal with what is good, what is ethical, and we'll have a free discussion on that. So to start off, we have Dr. Devan has introduced our panelists who are here to enlighten us today. But before that, just a brief introduction to what we are going to talk about. So now we all know that there are two things. There is peptides and there are small chemical molecules, which we usually know as tablet form. Like you may have your glimepiride, but they will be generics. So they are easy to copy. But peptide chemistry is little difficult and it's a biosimilar which can come out of this peptide from the originator molecule. So we know that this peptide is very important for semaglutide. So let's look a little bit into that. So there's nothing much to say about the growing burden of diabetes and weight in India. It's all around us. The recent INDIAB study showed that about 101 million adults in India live with diabetes, and almost a similar number live with prediabetes or impaired glucose tolerance. What comes out astoundingly is 254 million live with generalized obesity. That's almost double of the diabetes, and you can well understand that we are definitely sitting at an epidemic of all the complications associated with diabetes and obesity. And in our clinics, especially for me, compared to what I used to see 30 years back, 90% of people who come with diabetes are battling their weight issues. It's so difficult for them to lose weight for so many reasons. It could be drugs they are taking, their food habits, or just their psychosocial temper that they are in. So proteins and peptides have been forefront of metabolic care. We all know that. Right from insulin 1920s, when insulin was discovered, it was such a breakthrough molecule. And we had the Banting Best and the Nobel Prize for that. Much later, around 2000, 2020, we had another breaking molecule, GLP-1, which both of these are proteins and peptides and help first in diabetes control. And now with GLP-1, we have so many added benefits. Apart from weight loss, they seem to be good at every cellular and molecular mechanisms we can think of, right from Alzheimer's and so on and so forth. So they are two important breakthrough molecules, I would say, that have come out in diabetes and metabolic care. Now, this is the line of discoveries. So 1922, we had isolation of insulin, and thereon so forth, we had recombinant insulin somewhere in 1980s. And then, of course, we had in 2000s recombinant factor 7A. And then in 2020, we had GLP-1s, where there was rDNA technology in Ribelsis, which is the oral semaglutide, or Ozempic and Wegovy. Ozempic is generally the same molecule as an injectable semaglutide in a smaller dosage, which can give in for any diabetic, and Wegovy was majorly looking at weight loss, and even in a non-diabetic. So it's a lot of experience in a difficult atmosphere. 40 years of expertise in rDNA technology. So Novo Nordisk is definitely a pioneer in this GLP and peptide technology, but it was also the first company to put peptide in a pill, which was Ribelsis. And it is really difficult because they get digested by the gastric enzymes. So it's difficult to make a peptide, but they were able to, with the help of some attachments called the snack. Now, this innovator semaglutide is available, like I have told you, in oral and injectable forms, and also discussed the Ribelsis for oral form for type 2 diabetes, Ozempic for early management of type 2 diabetes, and Wegovy for a chronic weight management. So, with this brief introduction, I also have to tell you that there is difference between the originator biologics, biosimilars, and synthetic copies. So original biologics, rDNA technology, Ozempic, even insulin for that matter of fact, it was there. And then we had biosimilar. Biosimilar are similar but not the same. And however, they can get an interchangeability designation, which makes them slightly superior to even general biosimilars. But synthetic copies are not, they are loosely called biosimilars, but they're typically not. They're smaller molecules and they're not necessarily identical to the innovator, and they're produced or by a completely chemical process. So all injectables that we are having now of semaglutide are synthetic copies. So is the synthetic copy same as Ozempic or Wegovy? The molecule may be somewhat copied, but the process is definitely not the same, because that they may not be able to have the knowledge of or be allowed to use. So usually they have a reverse technology to get to the originator. So with that introduction saying that it's highly complex, we'll hear from our esteemed panelists about more in detail about each and every aspect of the differences and the advantages and disadvantages. So to start off, I have the first question to Dr. Lakshmi Lavanya. How does rDNA technology positively impact the final semaglutide end product? You're muted, Dr. Lavanya.
Yeah. Good evening, everyone. This is Dr. Lakshmi Lavanya from Hyderabad. Thank you, Madam Ma'am, and thank you, Novo Nordisk, for arranging this meeting. Thank you, ma'am, for setting a context background for this very nice program where you're trying to explain to the audience and us the differences between the synthetic copies, I would say, and the original rDNA molecule that we have as semaglutide in various usable forms in both injectable and oral. So how does rDNA technology positively impact the final semaglutide end product? I would say the rDNA technology is very ancient, long in the process over years. This is in order to bring about a very good quality, I would say a pure molecule, a purity, and also consistency in usage to get the same results in terms of efficacy and safety profiles of this molecule in terms of a much more of an analyzed pharmacodynamic and pharmacokinetic properties. Consistency is what we are looking for as a prescribing physician and the using patients out there. also to maintain the cold chain in order to keep this molecule viable from the manufacturer point of view to the usage point of view to the patient. So it's a four pillared rDNA technology, which is basically a time-tested platform to make them. So the process involves a gene. The gene of the interest is identified initially, and then it is cloned, so to be able to multiply it into a larger usable platform. So the clone gene is again transcribed into a DNA vector, and then it's transfer the plasmid into the host cell in order to multiply it with cell culture and fermentation. So down the stream, there's a lot of downstream processing involved in terms of purification, clarification, filtration, and then the purified drug substance is formulated and bottled or like packaged, and again this formulated product is ready for use, and it is again further transported into the with a very careful cold chain in order to reach the consumer. So next slide, please. I have a few more things to explain. Is about the process defines the final product. When the peptides are involved, basically because it's a rDNA process, it involves about 50 years of research background in order to bring a very low intrapeptide fibrillations or variations in the consistency of the molecules purity. So it ensures a high purity and also low immunogenicity, which is again antibody formation, auto antibody formation, or antibody formation against this foreign molecule. So they should be low immunogenicity and also consistent immunogenicity in order to have a greater acceptance, which translates into a biochemical usability of this molecule over both intra-patient and inter-patient efficacy consistency should be guaranteed to be able to use this molecule in large number of people. So rDNA technology also assures the consistency in terms of all these qualities and gives the persistent quality or every batch to batch variation production, in different years of manufacturing, and the expiry date and all that has to be placed in the right place to prove the safety. So it basically turns into the final patient safety and efficacy goals has to be reached here. So that is why it is, I would call it a process that can be trusted by the prescribing physician. Next slide. So how do we differentiate? So is that what is my question on? I hope I answered
Your question has been answered.
Yeah.
Thank you, Dr. Lakshmi.
I'll wait for the audience to maybe have more questions on this particular question of yours towards the end of the discussion. Thank you.
You have one more question from me. So after the end of it, we'll have the Q&A sessions
Yes.
from the audience. So Dr. Lakshmi has very nicely taken us through how difficult it is to go through the whole process. And she has also emphasized it's just not the efficacy, but the safety also, which is so very important, and the immunogenicity. I think these are three terms of what Dr. Lakshmi has spoken that we should keep in mind. These are very, very important terminologies. So the next question is for Dr. Joe, and he would explain to us how do differences in manufacturing impact clinical outcomes. Dr. Joe? Have we lost Dr. Joe?
Joe, you're muted.
Thank you, madam. Thank you, madam, for the question. Yeah, definitely the manufacturing process, especially in biologics, the process itself makes a lot of difference. The final product may be, see, we know that peptide molecules, proteins, they have the primary structure. Can we have the next slide? Yeah, they have the primary structures, secondary structures, tertiary and quaternary folding, and all those things. And sometimes it is the way you manufacture itself can make the things different. That's what the science tells us and the theory tells us. So there have been a few previous examples also with molecules where the initial manufacturer product versus the further products, there was differences in the immunogenicity, which has seen with previously. I think the first episode happened historically with erythropoietin, where initially the manufacturer, when it became off patent, the other molecules came in, but there was difference in immunogenicity which was noticed, which later became a problem. But later on, people have started recognizing this as a problem, but that theoretically still remains, that the manufacturing process and the final peptide, though may be identical, there could be other things that may be affecting the immunogenicity profile, like the impurities. So in this case, with semaglutide, there is actually a paper which has come out, which is an original research article, which looked into the impact of manufacturing process and compounding on properties and quality of follow-up on GLP-1 polypeptide drugs. In this study, they looked and analyzed 16 samples of injectable semaglutide copies, 8 oral semaglutide copies, and 2 liraglutide copies, both manufactured via synthetic and the biologic process. And what they found was that some of them had unexplainable higher level of impurities. As you can see in this diagram, you can see that the blue lines are the originator semaglutide, and the red trace is the one of the 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 copies that has been made of semaglutide. You can see that there is a difference in various impurities that are there, some of them in quite a significant level, and some of them, and also the oxidative patterns. This is LCMS graph, which can, from this you can see that the unknown impurities found is significantly there in some of the one of the sample that they're showing. Can I have the next slide? Yeah. So synthetic copies tested versus innovator in this article, actually, depending on the various products, were found to have different kinds of impurities. Some of the impurities, which, see, it's not being said that the originator doesn't have any impurities or other manufacturing elements there other than the molecule, but those have been time tested and also been tested through the rigorous clinical trials and found not to be harmful in human use over the last so many years. So these new drugs which have been produced, some of them have been found to have impurities which are not there in the innovator, which would actually can, at least some of them can potentially be harmful. We don't know whether it can be harmful or not, because some of them need not be harmful, but there is always that risk factor that some of these impurities can be significant. And as Dr. Lavanya has said, some of them may lead to increased antigenicity or immunogenicity, and they can lead to increased oxidative formation, and maybe how much they would affect the stability of the molecule, and how the structure, the other contamination, whether these impurities can act as contamination, and whether these, when it is stored, can lead to oxidative damages or can lead to unpredictability in efficacy. And these things we don't know. We cannot say that it can be harmful right away, but these are things which can be potentially of a problem, which may come into face as an important issue. In the same way, next slide, please. Yeah, the madam has already pointed out the importance of SNAC when you look at the oral semaglutide molecule. This is the first molecule which has been made available in history of pharmaceutical history as a peptide that is available, which is bioavailable orally. We were all surprised by the way they made it, and you could see from the way the persons who are using it, we can know that it has been so difficult, because since decades people have been trying to get the insulin into orals, and a lot of research has gone into that, and a lot of money has been gone wasted in the waste bin, because people knew that having an oral insulin bioavailable could be a path breaker and could be the next mega molecule that is there. But none of them has been successful. I think it's something like saying that gold is also a chemical, so why can't we manufacture it in lab? So people have tried since centuries, but not been successful in manufacturing gold. In the same way, this is another golden molecule that we have got in the market, that is through SNAC. That is a side, the SNAC, they have made that peptide molecule bioavailable for oral consumption. But again, there's been a lot of difficulty, which the innovator has pointed out, that the right dose of SNAC is very essential for the bioavailability and absorption of the molecule. have always insisted that you should not break the molecule and give it, and that the same thing they have shown in the diagram also. Here you can see that 5 mg of oral semaglutide with 3 mg of snack versus 5 mg, the same oral semaglutide with half the dose of snack was tried. And you can see that the half the dose was totally ineffective in the biologically. It was not getting absorbed and the bioavailability was very low. So we would say that, okay, you need more snack, which would give the better efficacy and bioavailability. So the next study actually shows the same semaglutide, which has been combined with 300 milligram of snack and also with 600 milligram of snack. And again, you can see that the higher dose of snack is also reducing the bioavailability rather than enhancing it, or at least maintaining that with the lower dose of semaglutide. So the conclusion that the innovator has put out is that probably this is a molecule and this is a mechanism where the correct proportion of the both semaglutide and snack has to be there so that you get the maximum efficacy of the drug in the final better outcome for the patient. To the next slide. I think, yeah, that's so. Thank you, madam.
So thank you, Dr. Joe. I think another very important point, Dr. Lakshmi talked about efficacy and safety, and we have other confounders like reliability, reproducibility. Is it possible with the compared to the originator molecule, will we be able to do it in the synthetic copies that we have? I think very, very important points that we should ponder over the importance of. So the next question is to Dr. Ganesh. Where does innovator semaglutide stand in terms of clinical trial evidence versus semaglutide copies?
Thank you, madam, for that question. So actually, there's a huge difference between the innovator semaglutide and the copies as far as the evidence is concerned. We can see in this slide for the innovator semaglutide, initially they underwent the characterization study, whether the semaglutide, whether it is the same as the original GLP-1, then the preclinical study in animal models, whether it is safe in animal models. First it was seen whether it is efficacious and safe. Then came the phase 1 studies. In phase 1 studies, usually the around less than 100 volunteers are selected, and it's mainly to look into the safety aspect. So the various doses of the molecule, like in this case semaglutide, was given, and to find out whether there is any alarming side effects, and then whether it is able to reduce the weight as well as the glycemia. So that was studied in phase 1. Then the phase 2 studies, again there are two aspects, phase 2A and phase 2B. Phase 2A, it's mainly the to confirm the efficacy part, whether it is really efficacious. Then whether the dose finding study in phase 2B, they try to find out the various doses. And then finally the phase 3 trials, in which various trials are there. I'll come to it in the next part of my slide. So in that, they compared with the existing standard therapy to see whether there is any major difference and whether it is really superior or non-inferior to the existing standard therapy. So innovator semaglutide has gone through all these studies, initially preclinical animal models to phase 1, phase 2, phase 3. Huge amount of patients have been tested in various phases, especially in phase 3. Large number of patients, in every trial around 5,000 to 10,000 patients were tested, and it was found that it is very safe and very efficacious also. Whereas the copied semaglutide, what exactly they have to do? In fact, it is not biosimilar, it's a generic molecule because it's synthetically prepared. So it's a characterization, only the peptide, whether the same amino acids are there, that they will be finding out. And there will be then bioequivalent studies, whether the bioavailability as well as bioequivalent studies, only these three will be conducted. And they have to show that it is 80 to 120% equivalent to the original molecule. So they have to show that the HbA1c reduction is either more than 80% or less than 120% of the innovator molecule. Then there will be phase 3 studies that also conducted by only in a few hardly numbers, probably numbering around 100 patients. It will be studied in phase 3. So we can summarize that there is minimal testing, no phase 1 and phase 2 trials, directly jumping into phase 3 with the bioavailability and bioequivalent studies, and a very small scale short-term phase 3 study in limited subjects, and that too in limited indications, and done only in India. That too, all the companies we don't have, only few companies have this phase 3 studies also. And there are only 3 manufacturers in India as of now. And one company has done very short phase 3 study. And whereas innovator semaglutide is approved in more than 75 countries due to the robust and extensive testing done across various indications and countries and populations. Whereas these our copies, synthetic copies, are tested minimally, as I told, in only in minimal phase 3 studies and approved only in India as of now. Next slide, please. So there is a huge difference between the innovator and the copied semaglutide regarding the trials. Next, previous slide.
Sorry.
Previous slide. I want to discuss regarding the various trials regarding the innovator semaglutide. It has got wide range of clinically proven benefits across large phase 3 trials, which none of the generic semaglutide is having. So we can see in this slide various trials done by the original semaglutide. The STEP trial, in which mainly the weight loss was aimed. So 18% weight reduction was seen with STEP trials, and sustained trial, once again the glycemic control, it was studied, and up to 2.8% HbA1c reduction was seen in sustained trial. And in select and flow trials, approximately 19 to 20% reduced risk of all-cause death was seen. All-cause mortality was reduced by 19 to 20%. And in Essence trial, in which MASL was the main target, whether there was any improvement in MASL liver fat reduction, there was 37% improved fibrosis and 63% MASH resolution was seen in patients who were using semaglutide. And primary prevention trials, the SOUL trial has shown clearly that it is useful in preventing cardiovascular death, and it is used as in patients who are not having atherosclerotic cardiovascular disease risk factors. So that is the reason CDSCO has approved semaglutide for the primary prevention also, for the original innovator semaglutide. And as far as the cardiovascular outcome trials are concerned, we have a lot of data. The studies like SOUL trial with the oral semaglutide, SELECT and SUSTAIN 6 with the subcutaneous semaglutide have shown reduction to the tune of 14 to 27% in major adverse cardiac events. And STEP heart failure trial with preserved ejection fraction showed a reduction in 69% reduction in heart failure events or cardiovascular death. And whereas as far as the kidney outcomes are concerned, the FLOW and REMODEL trial has shown 24% reduction in major kidney disease events. So suffice to say that the original innovator semaglutide has lots of data regarding the reduction of cardiovascular mortality, reduction of HbA1c, reduction of weight, and reduction in the progression of diabetic kidney disease. Whereas the generic semaglutides are just hoping that it will be mimicking the innovator semaglutide.
So,
yeah, next slide.
okay. So thank you, Dr. Ganesh. I think you have emphasized that looking at all this data does give us confidence that this molecule, originator molecule, is definitely different. And I don't think in evidence you can just extrapolate just because the molecule seems similar, and you need this data to confirm that it is as good. So without this data, we cannot say that the synthetic molecules also will have the same benefit as the originator molecule. Thank you very much, Dr.
Ma'am,
Ganesh.
it's like me. I'm Dr. Ganesh. I'm having 2 hands, 2 eyes, 2 ears. I also want to become Dr. Maladhar Malingam.
Yeah, or me, you, either which way. So, Dr. Lakshmi, your next question is, do
Yes.
synthetic copies come with the same regulatory approvals as with innovator semaglutide? I think a very important topic for the delegates. So please, shall I go to the next slide or?
Yeah, thank you, ma'am. So that's a very good question because first we need to understand what are the marketing indications or basically clinical indications for using semaglutide. So it comes in 3 forms, as we all know. Ozempic, the weekly 1 semaglutide injection, is primarily approved for first line for type 2 diabetes as an adjunct to diet and exercise. So this is a very important take-home message that it is first mainly for diabetes. Many people are just using this for obesity, and so many of them are having a trouble to use it for a normal BMI diabetic patients, where it is very much needed. I mean, or a slightly high BMI. So first line of indications, type 2 diabetes, to improve glycemic control in adults with type 2 diabetes, where you can also use it before metformin also. Sometimes you might find the place is very rightly found for this molecule even prior to metformin. So feel free to use it freely and also early in terms of their onset of diabetes, because it's much used to give life to beta cells to the pancreas, and also has no hypoglycemia, and a very robust molecule in terms of HbA1c reduction, keeping in place the safety parameters. So second thing, to reduce the risk of major adverse cardiovascular events, so it comes into the leftmost vertical column in ACE and ADA guidelines for diabetes, where GLP-1, I mean, as the GLP-1 RAs are coming into the leftmost corner. In that, semaglutide is preferred because of all the studies, as Dr. Ganesh just mentioned over the previous slide, had a complete array of the study results across phase 3 pivotal trials across the entire world with so many subjects in place. So to reduce the risk of major adverse cardiovascular events in adults with type 2 and established cardiovascular disease. Hence, this has been initiated by many cardiologists, or sometimes co-prescribed by our cardiology colleagues, and they give us a call and ask us, please consider this molecule for this purpose. Additionally, also to reduce the risk of sustained EGFR decline and ESKD risk and CV death in adults with type 2 and chronic kidney disease. So that's why we also get a call from our nephrology colleagues, or sometimes when we are actually making rounds on a patient who needs this to protect the other organ failure here, or to reduce the progress of the chronic kidney disease, and it is fastly progressive in that particular patient profile, case-based management. So the prescription is coming from other specialists also. So these additional usages of semaglutide has to be kept in mind, because these are the on-label clinical indications for Ozempic in India. So yeah, next slide, ma'am. So this is again a broad range of on-label clinical indications for Wegovy. The second, Wegovy is the same Ozempic semaglutide in a higher dose, where the marketing indications go up till 2.4 mg weekly once, primarily indicated for weight management in adults as an adjunct to reduced caloric diet and increased physical activity for weight management. So here we see a higher dose of semaglutide. The patients get started on the lowest dose again, and then the dose builds up, but the name changes to Wegovy as the dose goes above 1.7. Additionally, also it has been used to reduce the risk of major adverse cardiovascular events in adults with obesity or overweight and established cardiovascular disease. And we need to pay attention that this is a group of patients where we're talking about obesity and cardiovascular risk without having diabetes on board, may or may not have diabetes on board. So approval is again weighted for MASH and MASLD, because the studies are over and the results are also almost ready, but the FDA approval is still awaited to treat patients, adults with metabolic dysfunction associated steatohepatitis, MASH, with moderate to advanced liver scarring or fibrosis. So that will be another indication. So we need to keep that in mind. Next slide, please. So again, the oral semaglutide Rybelsus is again, I would call it a game changer and a life changer. As Dr. Joe was mentioning, the preparation niche in it, the snack involved, and the amount of snack to make it the most absorbable with the least dose to make it the most efficient and the most safe. So that there was the catch. So that was a very, very amazing, I would call it an amazing discovery of a peptide that has to be injected, has been brought into the market as an oral semaglutide. So this is one of a kind even now. And Indians are lucky to have Rybelsus in India, because this is not available in the US. So many patients there have missed the use of Rybelsus in the US. So I would definitely call it a life changer and a game changer, and primarily indicated for, of course, diabetes, to improve the glycemic control in adults with type 2, and adjunct to diet and exercise, also as a startup or addition wherever there's a need for a GLP-1 RA in terms of their glycemic regime. So additionally, it can be used to reduce the risk of major adverse cardiovascular events in adults with type 2 who are at high risk for these events, where this is the only OAD to have an open on-label indication for primary CV protection. So that again is based on all those pivotal CVOT trials, as mentioned by Dr. Ganesh, the one slide prior to my question. So next slide, please. So I would call it synthetic copies have limited regulatory approvals because it has not been studied in so many pivotal trials. The CVOT trials were not taken place. RCTs were not done on the phase 3 or even the preclinical phase 1 and 2, which are very, very important. And again, so much data, so much publication, so much review articles, and so much usage was not there, of course. So there's a very limited approvals for the synthetic copies. It is approved to improving glycemic control in adults with type 2, used alongside diet and exercise in combination with other antidiabetic agents, or as monotherapy if metformin is unsuitable, or even you could try it prior to metformin, is what they say, just like any other good GLP-1 RA. Additionally, for a few copies, it has also been established as a regulatory approval has been taken for chronic weight management in adults as an adjunct to reduce caloric diet and increase physical activity for weight management. But there is no on-label indication for the long-term benefits, especially in terms of the CVD, the CKD, and the MASH, etc., where Ozempic even takes a leap ahead than the more latest tirzepatide also. There are some awaited reports from regarding the CVD or the MASH, etc. So Ozempic being the most used GLP-1 RA in the latest generation of the GLP definitely takes a stand here. Yeah.
Thank you, Dr. Lakshmi. I think those are very, very valid points, especially the peptide in a pill. So it's really beneficial for those who are scared of a prick. And definitely these data that you mentioned, no? All the Essence trial, especially such outstanding data in Essence trial. First time we had some drug which was looking at fibrosis, no, of the liver. So
Yes.
I think without that kind of data, difficult for us to think of other molecules. So the next question is to Dr. Joe. How important is cold chain compliance and device convenience in a country like India? I think, again, a very important question.
Yeah, madam, definitely very important question. What I would say is that, again, I will go back to the insulin. I've heard experts and even the company persons, the scientists in the various forums talking about, when you talk about insulin, the most important thing is the cold chain. The manufacturing process is definitely there, but the manufactured process in its same bioavailability and its same purity reaches to the person who is taking that injection is the most important thing. It is just very easy to talk about the importance of cold chain to a person who is a non-vegetarian, because when you eat any non-veg, say fish, you catch from the sea, and the time that it takes to reach to your table, and how much the way it is stored, how much difference it makes, not just in the taste but also for the tummy, is something and every non-vegetarian would come to recognize very fast. But for vegetarians, it may be difficult. So the actual cold chain, and also I don't remember who told me that, because again somebody related to the industry high level told me that the cost of insulin predominantly is the cost that we spend on the management of cold chain. The actual product cost is much less, and we spend very less on the manufacturing process and the manufactured product, but we spend a lot of money and lot of effort in maintaining the cold chain. So cold chain is one of the most important aspects to be maintained. And there, one important thing that we have to understand is that the innovator semaglutide is being cold chained by a company which knows how to cold chain, and they are the pioneers in cold chain maintenance, along with the Novo Nordisk is a pioneer with Eli Lilly in insulin since decades, and their experience is something that can be unmatched in cold chain maintenance. So it's and semaglutide also is a temperature sensitive drug which requires cold chain maintenance and maintenance of temperature between 2 to 8 degrees Celsius to preserve the integrity and clinical potency. And that fridge to factory to fridge, that cold chain maintenance, nobody can doubt that of an innovator. And so many new people coming up how they can maintain the cold chain is something that even before this presentation I was worried, because so many manufacturers with no experience, they can just buy the product from the person who is manufacturing it, but how to get it branded and reach it to the customer with maintenance of the cold chain is something that has to be really a concerning thing, which would be answered in the next few months or one next year, I suppose. And second thing is the integral the device, the flex touch. Flex touch is something that has come with the insulin, and the patients who have used it find it so convenient. It is, and they have transferred the same technology of flex touch to semaglutide, the innovator. It is a very simple to use, and the push button is not there, so you can just have it very easily with very low injection force. And the patients who have used the older pen versus the flex touch pen have always said that the easiness of use, and the company, and also we are also confident of the drug delivery and the precision of drug delivery. So we have recognized that this is the newer generation of the drug delivery system through the pen is the flex touch pen, and semaglutide comes in the flex touch. Thank you, madam. Madam, you are muted, madam.
I couldn't agree with you more on this cold chain. It's so, so important. I still remember when they sort of switched and they had the local companies. The cold chain was not in process. So they reached it to people who, in places, even those remote places where there was no cold chain. And we used to see sugars going all over the place because they were not working at all. So I think that is such an important point, and definitely devices makes life easy for everyone with the newer development of these easy devices. I think both are very, very important for us. So the next question goes to Dr. Ganesh. And what is the level of real world use or experience with innovator semaglutide?
Yes, madam. So actually, the innovator semaglutide we have been using since the last 4 years with as far as the oral semaglutide is concerned, and subcutaneous semaglutide they launched recently, or one almost 6 to 8 months back. So the Indian usage, we have about 4 years of usage in India for oral molecule Rybelsus oral semaglutide, and if participation of trial in the trials, approximately 3,500 or more than that people are enrolled in these various semaglutide trials, and Indian users approximately more than 4 lakhs. 4 lakh Indians are already using it, and in India now both injectable and oral formulations are available. Injectable also, both Ozempic and Wegovy are available since the last 6 months. And if the global evidence, if we look into, it's a large data actually. It's a they got the global approval in 2017, and approximately 50 phase 3 clinical trials were conducted, and 60,000 trial participants. 60,000 trial participants, and more than 75 countries it has been approved, and for more than 49 million person years of exposure is there. This is a global evidence. Nobody can match this actually. Huge data if we look into the global population. But Indian perspective, since it was launched the only 4 years back, the oral one, and the subcutaneous was launched recently, we have approximately 4 lakh Indian users. It has shown a good promise in the real world.
So though we have less in actually coming, even we were part of those global research. I was part of Sustain 6. So even our patients go into that pool, and whenever a molecule is introduced, we also want to know how it works for our Indian population. So I think even the sub-studies are there on Indian population, which seems to be very, very good. Thank you, Dr. Ganesh. And then we have one question for all the panelists. Do you see a clinical need to switch from innovator semaglutide to a copy? We'll take Dr. Lakshmi, and then we'll go to Dr. George and we'll come back to Dr. Ganesh.
Thank you,
Yes.
Dr. Mala. After talking about the entire session, this question has been posed. So we've been debating on using the original molecule because of the extensive rDNA technology, the research, the safety, the efficacy, the indications, the so many very good trials and the results, and the use of the use of the cold chain that has been kept in place to assure that the same molecule gets to the individual who's taking it as prescribed, as we want, as what we have researched, and what should be given to them in terms of the use of this molecule in terms of getting their sugars better. So in with this in place, I don't see a need of a synthetic copy, but what can you talk about how it, how it's the and the maya of the world, it just goes on and on. People prepare more, there's more use, there is more happening. So it's just the happening of the world which takes all these, it brings a variety. It's a variegated, multifarious, variegated world, and we'll have a lot more variety for our dispensal here. So the thing is here, we need more trials from the synthetic copies. We need a proper data. So we need the study participants to have used this comparing head-to-head trial, or at least a trial of the molecule synthetic copy, compare it with placebo, and do it for a longer time and have very good publications. Otherwise, I'll have to use it with the informed consent. Because even with the GLP-1 RA who have been researched so long, patients have different opinions and different views, and suddenly a side effect comes up, and then we have to advocate them and convince them, and again, it is such a careful management with these molecules overall. Now, when you talk about a synthetic copy coming side by side, I think it becomes much more complicated from the prescriber's point of view, because we only care that the patient should be safe and it should be an effective treatment. And not only that, but it takes over with the other comorbid illnesses. We don't want them to get more problems with something new happening here. So I would never switch from the innovator to copy. I would not do that. Definitely not switch, especially now because the prices have been also dropped for the innovator. And it has been dropped the second time, and it has been matched almost close. With so much variety, garden variety going on, we don't know how the every pen is different, every name is different. There's not one, there's already 10-15 names come into our offices already. And so their availability and the first of all, their availability, the prescription, how is it transferred across, and how is the cold chain maintained, and how are the results of these patients. So we don't want to research on our day-to-day clinic arriving patients. So I would be very, very cautious on this, and we need a lot more to see. I'm waiting and watching about what other doctors have to say, and what the trials have to come further up, and what publications and what the experts have to comment on these molecules, the copies, synthetic copies. I'm still waiting, ma'am, and I have not much to say.
Dr. Lakshmi is saying we don't want our patients to be guinea pigs. Let other doctors use, then I will use it for my patients.
But
Yes.
already it's a lot. It's a lot to be very careful, ma'am, with the existing GLP-1 RA. Patients say the 0.25 worked better, the 0.5 is not so great, my appetite has come back, or I'm having issues. Even with this, there's so much carefulness going on. And I can't imagine a synthetic copy on board.
Dr. Joe, your thoughts?
Yeah, madam. Here, I would go by the history actually. You would remember around, maybe around 20 years back, exenatide became off patent. 1 year it was withdrawn. Nobody was using it because the efficacy was not at all matching with the originator. And the company by themselves withdrew it. And then we have the experience with liraglutide also. There, the Novo Nordisk was very supportive for the generic manufacturers by withdrawing their product by themselves. So that all the doctors in India, including myself, who were actually using liraglutide in our patients, were actually compelled to shift over to the generic innovator. From the innovator to generic, because the original innovator was not available. I think they were actually priming us with the experience with the liraglutide, because we were compelled to shift to generics, and we found that the generics were not at all effective. And the patients actually came back saying that, doctor, this is not working. This is not going to do this thing. I don't think, why don't we go back to the previous one? Then I said, see, I didn't shift you just because it is cheaper. I just shifted because the other one was not available. That was the reason. But finally, I can tell you that all my patients who are on liraglutide, I had to stop because the generics were found ineffective. So if you ask me at this current point, I'm not planning to shift any of the innovator patients on innovator to the generics, because I have burned my fingers, because company itself have primed me with liraglutide that shifting to generics may not be as effective, or may or the other things that starting and trying to see. I may try it in a new patient, not but on I won't try to shift the patient.
I think I fully agree. As a way of thinking itself, it's not right to switch. I don't think we should be doing that, isn't it? So, Dr. Ganesh, any thoughts from your side?
Yeah, actually, I totally agree with Dr. Lakshmi Lavanya and Dr. George. There are a lot of issues in switching. The innovator definitely works better. We had the experience of both exenatide, liraglutide, and even the insulin. There is a difference between the innovator molecules and the copies. And cold chain is one of the biggest concern. So with all the generics, that will be the biggest concern, because they don't have any experience of handling the cold chain. All these while they were only manufacturing tablets, which were the cold chain was not at all required. Now everybody wants to jump into this GLP-1 area. So probably there is one more concern. And one more final last concern is a lot of confusion. So many pens, so many vials, so many the syringes. So even endocrinologists, we are dealing with only diabetes, and diabetologists who are looking after diabetes also totally confused. There are some multiple companies coming up with various pens. One with the 15 mg pen, one with 5 mg, one is with the 2.4, one is 2, and they are advocating 0.71, then after 1 2 mg, all these, which were no trials were conducted. 1 mg, 2, after 1 mg 1.5, then 2 mg, so which were trials were not conducted at all. So even such dosing is there in the generics. I don't know from where they got these ideas. So with all these confusion being around, I don't think it is worth shifting our patients to the generic version.
So thank you, Dr. Ganesh. I think all our three panelists have come out very clearly and strongly that we don't have enough data with the synthetic copies. And definitely we should think twice before thinking, even if it is cost effective, we don't want to have patients getting into problems of efficacy, safety, or predictability of outcomes. So I think these are three very, very valid points they have talked about. And just to conclude, it's not moving. Have you taken the control, Dr. Devan? Dr. Devan, I'm not able to shift the slides. Are you there? So just to conclude, why are the synthetic copies not the same? We've already understood, I think the previous slide, please. The differences between them. That is, we have a huge heritage with the innovator. It's been there from 2017. The technology is rDNA based. Many studies and many participants assuring safety, efficacy. All our panelists have talked about it. And always I'm more comfortable when it is approved in more countries than just India. And that gives me a stronger level of confidence to say, okay, this drug is good. Just having it local, Korea or India, always makes me a little worried. Is it the good molecule? Storage cold chain has been emphasized quite sufficiently by all our panelists, and a device. The newer technologies towards better device is most important. And I think maximum is the patient experience of more than 49 million patient years. I think with that confidence, all of us are sure that we'll be very comfortable with the innovator compared to synthetic. We don't have much data, just recently approved. Even if we have some patients, it's just a handful, and none of them have come back to us for any kind of follow up. So we don't know. So unless we have enough, we have to wait, be patient, don't jump the line to just for a few pennies. I think that is not the right way to go forward. So for quality, purity, consistency, and cold chain, which are very important factors, I think we should stick to the originator till we have more data. And last, but the dosing has already been told to us by Dr. Lakshmi. So I won't go to it in much detail, especially since we have come to the end of the program. But just to say, Ozempic, like she rightly said, is for every diabetic, irrespective of their weight, can go start from 0.25, increase it to 0.5, and then go up to 1 once a week. Wegovy is the same, but you can go up to 2.4 once a week. The weight loss, usually they recommend from 1.7, but again, because of tolerability, it is better to start slow and build it up over a few weeks. And Ribelsis, we all have been using. So start with 3 mg, go to 7 and 14, at least, and exactly the way they tell us to take it, early morning with 120 ml of water, so that there's no interaction. And these are some very important points. So now if there are any burning questions, Dr. Devan, you can ask the floor. We are almost at the end, 9 o'clock of our meeting.
Yeah. So are we open for some 1 or 2 questions?
Yeah, very quick questions. You can ask the panel and we'll wind up.
Sure. So for Dr. Ganesh, given that originator semaglutide has a robust CVOT data, like sustain 6 trials and select trial, how should clinicians interpret efficacy claims of follow-on semaglutide without such long-term outcome data?
Yeah, I think since they don't have any original data, they are just hoping that they will be having the similar outcome with the as like the innovator, which is not correct because they are totally the manufacturing process are totally different. Whether it will work similarly, we don't have. So probably we can't extrapolate the data from the select or sustain trials to the synthetic semaglutides. I don't think it is possible. So probably. The message should clearly go clearly that the sustain and whatever the select trials are only for original semaglutide, not for the generic ones.
Sure. Any other question, Dr. Devan?
Yeah, one last question to Dr. Lakshmi. If a patient experiences unexpected GI intolerance or reduced efficacy after switching, how do we differentiate between drug-related issues versus patient-related variability?
I don't know. It's a hard question. Because you're asking for a switch after they have been switched from the original originator molecule to the copy. Then if there's a sudden GI issue, is it related to the switch of the molecule or is it something else? Or, you're trying to ask me. Is that right? Did I get the question right that way?
Yes.
Yeah.
Yeah, so it's a hard thing to say, but since you have switched the molecule, is it the same dose or you've gone up to the dose, and what is the reason? What is the other comorbid illnesses? How is the basic GI health of this person? Is there any additional food intake that could have maybe a new germ like a gastroenteritis coming along? We have to look at the patient. It's a very, very personal case-based approach that we'll have to look through, and we have to go by what is the best approach, best clinical management approach to give a relief to the patient. And it's very hard to say that the switch of the molecule can increase the GI side effects. I don't know. There's so many things here: the efficacy, the safety, the is the right pharmacokinetic, pharmacodynamic approach going to this patient after the switch. We don't know, isn't it? We don't know what the switch does. We just know the synthetic copy as a separate thing. We know the originator as a separate thing, but we have not known the switching from one to the other. What can it do to the body systems in various of the drug effects? So it's a very hard question to answer. I would say it'll be an extrapolated some blabbering if I say something.
Don't have too much experience with this newer
Yeah.
molecules, no? Just
Absolutely.
it has started, so we really don't know.
Yeah.
So, Dr. Joe, any last thoughts?
No, madam, as always, only time can say what's going to happen.
Absolutely.
But history actually points the history that we have from exenatide to liraglutide actually is not very positive for the generics that is coming in, but only time can say.
So I think what we have to conclude is we need much more data than what is presented to us. And even for all the delegates out there, just don't start just because a few company guys come and terrorize you. You have to look at data. Ask them for the phase 3 trials, ask them for the bioequivalence, bioavailability, immunogenicity studies, and then only if you feel convinced, tell the patient frankly, this is all the data we have. Because now I also find patients are coming and asking me, Dr. Mala, I believe there's a cheaper version, shall I take? So you have to explain to them and put the onus of responsibility on patient itself. See, it is there, but we don't have much data. If you want, you can try. So I think with that we have, because that's our responsibility. We can't just be shifting indiscriminately because we don't know outcomes ourselves. So I think with that, I should thank all the panelists who were so excellent, Dr. Joe, Dr. Ganesh, and Dr. Lakshmi, for having joined us, and handing the session back to Dr. Devan for his conclusions. Thank you very much and good night.
Thank you, Dr. Mala. Thank you, Dr. Joe. Thank you,
Thank
Dr.
you,
Ganesh,
madam.
sir.
Yes, Dr. Devan, any final words?
Yeah, just the closing remarks.
Sure, go ahead.
I like to thank you all. I'd like to thank Dr. Lakshmi, Dr. Ganesh, Dr. Joe, and Dr. Mala for coming and giving your thoughts on this originator semaglutide. Thank you so much for giving your precious time. Thank you so much, and thank you all the delegates for attending this webinar. Thank you so much. Have a great day. Have a great night.
Good night.
Thank you, madam. Thank you, Joe. Thank you, Dr. Lakshmi. Good night.
Bye.